Good evening, grade 11. I hope you're doing good. Today I'm going to continue what we explained last time about the structure and the chemical components of chromosomes, okay? Now, we said that chromosomes are best visible at metaphase because they are in the condensed state, okay? Now, DNA identification by Fulgen reaction. We spread the cells on a slide. We put for it hydrochloric acid at 60 degrees Celsius for 15 minutes. Then we rinse it with water, okay? We add Gibbs reagent. We can see the DNA colored in red. Look here. Okay. Here on the inside, we can see the chromosomes, okay? The genetic information. Uncoil the chromosomes, okay? So, we said previously that the chromosome is made up of two chromatids joined by a centromere. We said that the DNA is long, but how does it fit inside the nucleus? Because it becomes condensed. How does condensation takes place? Okay, I'm going to refer back to the information that I gave you, then I will continue explaining here. We said, guys, that condensation takes place inside the nucleus, okay? The DNA rotates around the proteins that we call histones. Look here, okay? This is the chromosome that is made up of two chromatids. As we can see here, okay? This is the genetic information, this is the DNA. Here, what happens? The condensation, okay? Here, condensation of chromosomes takes place. How does this condensation takes place? In the presence of a proteins called histones, okay? Can you see these bubbles? These small bubbles, okay? These are the histones. They wrap the protein in a way. I showed some pictures to you in class. They wrap them, okay? They wrap the protein so that the protein that is the histone wrap the genetic information, so it becomes coiled or condensed inside the nucleus, okay? Now, as we said that the protein with the histone, the protein that is the histone with the genetic information, we call them nucleosome, okay? So these are nucleosomes. This is the DNA. It's being coiled with the histones. DNA with the histones are referred to as nucleosome, okay? Now, structure of DNA. In 1953, Watson and Crick suggested a structural model for DNA. As we said previously, the DNA is a double helix, okay, double-stranded. It is, which means that it is made up of two chains, two strands, okay, twisted, okay, not flat. It is twisted like this shape, okay. It is twisted. DNA has the shape, we say that, of a twisted ladder. Here they put a big ladder, the ladder that we climb on, okay? It is twisted, so it has the shape of a twisted ladder, okay? As we said previously, that when we are talking about the DNA, what is it made up of? We said that the DNA is made up of what? Nucleotides, right? It's composed of a sequence of nucleotides. And we said that nucleotides, each nucleotide is formed. Look here. Here we have the nucleotide. This is nucleotide A, this is T, this is G, this is C. So this is adenine, thymine, guanine, and cytosine, okay? A is complementary to T, always. These are couple, A and T, G and C. Okay, we cannot find A with a G or with C and vice versa. A always combines with T and G always combines with C. Okay, so A, T, G, C. These are the four nucleotides that we know. Each nucleotide, Okay, look here, okay. Each nucleotide, this is one nucleotide. This nucleotide is formed of what? G, that is guanine, okay? G, that is the guanine, يعني nitrogenous base guanine, deoxyribose sugar, and phosphoric acid. So, the nitrogenous base with the deoxyribose, with the phosphoric acid, these form one nucleotide, okay? So, this is C, nitrogenous base C, with D, the deoxyribose sugar, with the phosphoric acid, they form one nucleotide. This is another example, okay? This is another example. This is another example of a nucleotide, fine? Now, as we said previously about the DNA replication, we said that in the process of DNA replication, in which the DNA strand duplicates, and this takes place inside the nucleus during interphase, we said that the DNA replication is a semi-conservative method. What do we mean by semi-conservative method? This means that we have part of the new DNA or the daughter DNA, we have part of it old and part of this daughter DNA new. And I will explain this in more details, okay? Now, the initial DNA double helix opens during interphase.